mean, if you think about it, most cells on the body are really, really tiny, and these are much more expansive. They also have multiple nuclei, as we saw in the pictomicrograph. And then I mentioned on the other video, they have a lot of mitochondria to produce the ATP they need in order to continuously work for us. We'll especially look at that when we get to the cardiac muscle and AMP2. We'll talk a lot more about that since it's constantly contracting and relaxing. Also, they have glycosomes, which store glycogen. Glycogen, if you remember, is a big, large molecule of sugar bound together. And then when we need that sugar, we can break it down and put that into the blood, or in this case, into the muscle. Also, we have myoglobin, which is for storing oxygen. And then we have myofibrils, sarcoplasmic reticulum, and T-tubules, and I'll get into what they do for the muscle cell here in a few minutes. All right. There we go. So again, myofibrils are densely packed rod-like elements. It's about 80% of the cell volume for the muscle cell. So if we look at the muscle cell here, this whole entire thing, we can see that it's made up of these myofibrils. And these striations, the one you see under the microscope, are caused by these dark A bands and these light I bands. Now, when we zoom in further on this, I will show you those bands a little bit more in detail. But that's what gives it that striated appearance. You can also see the multiple nuclei. See one here, one here, one here, that are also characteristic of skeletal muscle. Remember this for your test. I will probably ask you at some point what is the smallest contractile unit or functional unit of a muscle, and that is the sarcomere. And it's made of many myofilaments of actin and myosin, and we're going to talk about them in detail in a minute as well. And then the region of a myofibril between the two successive Z discs is called the sarcomere. So we're going to look at it in sarcomere kind of blown up here in a minute, and we will look at the Z disc and then the midline or the M line. And then also we'll get into how actin and myosin work to help the muscle to contract. So the thick filaments are myosin, and then the thin filaments are what we call actin. And myosin run the entire length of an A band, and then the actin runs the entire length of the I band and partially into the A band. The Z disc, the H zone, the M line, I think it's just better if I show you these rather than just talk about them. So let me just flip on over to this slide. So up here you can see the sarcomere. And again, we have two Z discs, and these will move towards this zone in the middle here. So again, we can even expand this out further and look down here at the Z disc and then the M line. So again, they're going to pull inward from both sides, and that's going to shorten the sarcomere. When all the sarcomeres shorten together, that's going to shorten the entire muscle. Right? So we need to kind of understand how a sarcomere works and the sliding filament theory, and I'm going to post a video for you guys to watch on that instead of going in too much detail with that. I think it's just easier if you see it presented live where you can watch the movements and that type of thing. If you notice down in the sarcomere, we have these actin and myosin that we see here. So this is our actin in blue, and then our myosin is down here with the heads in pink. And these heads will actually bond with the actin filament and move it along. It's almost like a ratchet. You know what a ratchet is? Like you work on a car or something, but it moves in increments. So as these heads encounter the actin, it's going to start pulling this Z disc towards the M line. And again, I'm going to show you this. It's a lot easier to show you, I think, than just to talk about it. So just kind of keep in mind that's what the sarcomere looks like. We have the actin and myosin, and then they are going to pull against one another to pull those Z discs in towards the M line. So here we can see myosin. We have two interwoven heavy polypeptide chains, and then the myosin heads contain two smaller light polypeptide chains that act as cross bridges. And we're going to look at that cross bridge formation here in a minute. It requires ATP in order for this to occur, for the sliding filament to move past one another. So ATP is going to be involved, and we have enzymes, ATPase, to help catalyze that reaction to get this going. Now, if we look at the actin fiber, we have something called troponin and tropomyosin, and these are regulatory proteins that block the active site. So let me go back one slide. We're looking at, actually two slides. We're looking at this blue filament here, okay? And on there we have that troponin tropomyosin complex that blocks the active site that this myosin wants to bind to. So as long as it's blocked, the muscle will remain relaxed. Once we open those active sites, then the myosin heads are going to want to start to bond here and ratchet this along. Okay, so that's kind of what we're looking at. So let's flip back over here. And the active sites right here on this one are covered. All right. With the presence of calcium, this is going to move off these active sites, open them up, and allow the myosin heads to make contact. And that's when the muscle starts to contract. So the final trigger for muscle contraction is that influx of calcium that we're going to look at in the video later. Right. So here you can see our actin and myosin. Again, actin's in blue, myosin is in kind of the pink-red color here. Again, these are going to interact with one another and move these in towards the midline, move those Z discs towards the midline. Again, I think it'll be a lot easier if you watch the video that I post, and you can kind of watch this in action. It's a little hard to explain it without showing it to you. So again, just make sure you remember the troponin tropomyosin blocks the active site on actin. That prevents these heads from binding to that active site. But when calcium is available, these proteins, these troponin tropomyosin, will move off the active site, open them up, and then these heads will start wanting to bind with them. And again, this all requires ATP, so it's going to take energy to do this. Now, where does that calcium come from? Well, in skeletal muscle, it mainly is held in the sarcoplasmic reticulum, and that is shown in blue down here. And you can see that it's surrounding the muscle fiber. And once the trigger, what we call an action potential, comes in, it's going to move across the cell membrane, or the sarcolemma, and then down these T-tubules, down into the sarcoplasmic reticulum, and it's going to open up calcium channels and allow that calcium to flood the muscle cell. And when that happens, that's going to remove that troponin tropomyosin complex we were just looking at, and that's going to cause the muscle to contract. So again, it functions with the regulation of intercellular calcium levels. And again, it plays an important role in muscle contraction by storing that calcium and holding it until the signal comes in from the nervous system to open those channels up. Now, I know we haven't really talked about action potentials. We'll talk a lot more about them in the nervous system chapters later on. But I'm going to get into it with the muscles as well, because we need to understand how that works in order to understand how the muscle does contract. So an action potential simply is an electrical impulse that moves along, usually from a nerve, into the muscle, and then it's regenerated in the muscle and carried down to these little gates that open up and allow that calcium to flood the cell. Right? So just think of an action potential. And anytime I say that word, just think of almost like electricity running through a wire. Right? It's simply a signal that's going to go from the brain to the muscle via the nervous system, and then it's going to trigger those calcium channels to open and release that calcium onto the muscle. So again, the T-tubules are going to penetrate the cell's interior at the A-band and the I-band junction, and then it's going to, again, deliver that action potential to the cell. Right? So this is what kind of brings the action potential from the outside of the cell down inside so that we can open those calcium channels. So this is our T-tubule here, right? And we call these transverse tubules. That's where the T in T-tubule comes from. Right? So again, here's a muscle fiber, and we've kind of blown up everything so you can see it real well. So again, the sarcolemma is our cell membrane for the muscle cell, and then these myofibrils are all located together. And again, if we can contract all of them at once, we can generate a pretty massive force. Now notice down here we have a lot of these mitochondria embedded in here. Again, this is going to make the ATP that's required for muscle contraction. So again, just kind of keep it in mind that there are a lot of mitochondria that pumping out all this energy to the muscle to use. All right, so again, the T-tubules are going to conduct the action potentials deep into the muscle fiber. And then again, that's going to open up those gated channels that regulate calcium release from the sarcoplasmic reticulum. So again, it's that signal, that action potential, that gets the muscle prepared to contract. It's what's going to stimulate the muscle to open those calcium channels and allow calcium to bind to tropon and tropomyosin and induce the contraction phase. So contraction is simply the generation of force, and it doesn't necessarily cause shortening of the fiber. It usually does, but it doesn't always have to. We're going to go over the different types of muscle contraction here in the next video. So I think I'm going to stop right here. It's a good stopping point, and I'll pick back up here in the next video.